Okay, welcome back to General Virology, Chapter 1, Part 2. I want to talk just for a few minutes about possible origins of viruses, and then we'll delve a little bit into the history of the discovery of viruses, and lastly, talk a little bit about some other non-living infectious agents. Okay, so we don't really know where viruses came from, but there are these three major hypotheses: the precellular, the escape hypothesis, and the regressive hypothesis. And there's some evidence for and against all of these, so I don't know that one is really the favorite, or if there's scientific consensus for one of these. But I just wanted to make you aware. I think it's interesting to consider where viruses may have originated. So the precellular hypothesis, as the name implies, suggests that viruses predate cells. Um, this hypothesis presupposes that RNA was the first genetic material, and there's actually a large amount of evidence to suggest that is the case. Um, and according to this hypothesis, DNA actually first arose in viruses and was then transferred to primitive cells upon infection. And eventually, the RNA genome of the cells was replaced by this DNA genome that originally originated in a virus. Another aspect of this theory, or another hypothesis tied in with this precellular idea, is that the three domains of life, bacteria, archaea, and eukarya, may have actually been distinguished based off of these primitive RNA cells being infected with three different DNA viruses. So those DNA viruses were basically seeding the unique genetic starting material that was eventually going to give rise to these three separate branches of life. Now, the flip side of the precellular hypothesis coin would be the escape hypothesis, which says that cells existed before viruses. Viruses were actually once portions of cells that at some point gained this ability to escape and travel from cell to cell. So in this instance, in this hypothesis, you had these primitive versions of these cells from these three different domains that then gave rise to distinct groups of viruses. So you have, in the precellular hypothesis, basically viruses essentially helping to originate cells in their genomes. In the escape hypothesis, you have cells helping to originate viruses. And then the third, the regressive hypothesis, suggests that viruses were once completely independent intracellular organisms, so like analogous maybe to bacteria that might live inside of larger cells, but that they lost the ability to replicate independently, so are now intracellular pathogens, obligate pathogens. And, some of the evidence for this hypothesis points to the relatively recently discovered viruses, which stands for microbe mimicking viruses, is the part. And these are very, very large viruses, in fact, bigger than even some bacteria. And they do have some characteristics when infecting cells that look similar to certain bacteria. So, for example, they form these virus factories inside of an infected host cell. And there's a lot of structural similarities here, what's going on in the viral factories, to what happens when chlamydia bacteria infect a cell. But despite those physical similarities at the genome level, there's really not any overlap. If we look at the genes of the virus, compare those, say, to the genes of chlamydia bacteria, we don't see the similarity there. So in my mind, this is probably the weakest of the three hypotheses. All right, so let's go into just some of the brief history on the discovery of viruses. And we have to start the story with Louis Pasteur in the mid-1800s. And of course, Pasteur is famous for coming up with germ theory, or carrying out experiments that supported germ theory, which essentially led to the discovery of bacteria. And this whole idea that microorganisms are all around us in the environment, and some of those microorganisms can cause infectious disease. But essentially, the basis of his simple experiments was he had, a nutrient-rich broth in these swan neck flasks, and that broth would stay sterile as long as the neck of the flask was intact. Basically, that swan-shaped curve would prevent air from entering the flask. Whereas if you snapped off that neck, allowed air to get in, all of a sudden microorganisms would start growing in that nutrient broth. And so, it seems very obvious to us now, of course, okay, microbes were getting in with the air. But at the time, it was thought that, spontaneous generation may occur, right? You basically just spontaneously generate these diseases or these disease-causing microorganisms. But these experiments really showed, nope, it's in the environment, and if we can keep the environment sterile and reduce exposure to these microorganisms, may be able to reduce disease. Um, so that really led to this whole idea of microbes and microorganisms. And, even though viruses technically aren't organisms, they do get grouped into that category along with bacteria, basically because of their disease-causing abilities and the fact that they're very, very small. But the first virus that was ever discovered was actually a plant virus, tobacco mosaic virus. Was found in 1879 by this researcher Adolf Mayer, he was looking at this mosaic disease. You get these very characteristic symptoms of tobacco mosaic virus, this mosaic pattern on the leaf, hence the name, and necrosis, and some vein clearing. But he found that you could transmit that disease, that phenotype, just by transmitting the sap, or basically the juice from an infected leaf, to a non-infected plant. So showing that this was an infectious disease and not just some, environmental deficiency or nutrient deficiency or, insect pathogen or something like that. So that was 1879. In 1892, Dmitry Ivanovsky, Russian botanist, showed that the infectious agent could be filtered through a Chamberlain filter, which had been shown to remove bacteria, right? So a very fine filter. So that suggested this disease was not being caused by bacteria. In 1898, Martinez Biergenik, I'm probably butchering his last name here, um, showed that the infectious agent was still infective after being filtered through Chamberlain filter. And he really came up with this idea that instead of this infection being caused by a microbe, it was really what he called a living fluid, a contagious living fluid. So his thinking was the fluid in the plant had become infective, and once you transferred that to an uninfected plant, it was taking the disease along with it. So he actually coined the term virus to distinguish that from microbes, which was being used at that time to refer specifically to bacteria. And at that same time, two other researchers, Loeffler and Frosch, were doing similar experiments with an animal disease, foot and mouth disease, and they found that the causative agent of that disease could also be transmitted after being filtered through a Chamberlain filter, suggesting it was not caused by bacteria in a similar way. But they took it a step further. They tried an even smaller, newer filter called the Kitasato filter, um, and they found if they passed infectious fluid through that filter, it lost infectivity. And so what that meant was that this virus, this new word, this new infectious agent, was actually not a liquid, but a very, very small particle, right? It was capable of being filtered out. And so foot and mouth disease virus was actually the very first animal virus to be discovered. And it wasn't until 1900, another 10 years later, that we found the first human virus, which was yellow fever virus. You can see in this image, yellow fever outbreaks were pretty widespread. We had temporary tent hospitals set up in various parts of the South, predominantly, where yellow fever was a big problem back in the late early 1900s. And so here we've got a little timeline. I don't want to spend too much time on the timeline here, but just to point out some of these key events in early virology. And so, we were just talking about Pasteur, et cetera, and yellow fever virus, but we could actually take this all the way back to the mid to late 1700s, when Edward Jenner was basically coming up with a primitive method for vaccination against smallpox using fluid from cowpox sores. And of course, they didn't know this was, anything about viruses. That term hadn't even been invented yet. But we now know, that was a viral disease that they were working on, and they did have some success with these early kind of primitive vaccination attempts. And we can see discovery of some more modern uh, viruses, polio in 1909, HPV in 1907, found bacteriophages in the teens, 19s, herpes in 1919, etc. And so with this new knowledge that, okay, viruses are actually very small particles that can cause disease, um, if we think of this in terms of Koch's postulates, which had been established to try to, as this, this set of guidelines that would show that a particular infectious agent or microorganism was actually responsible for a specific disease. Um, there was this desire to try to apply this to viruses as well. And you've probably seen Koch's postulates before, but they said, you have to find the organism, or in this case, the virus would have to be present in every case of the disease. The virus would have to be isolated from the host that was diseased and then grown in pure culture in the lab. Then you'd have to be able to introduce that virus back into a healthy, susceptible host and find the same symptoms again, and then yet again be able to recover that virus. Right? So it's pretty rigorous. And this was actually very problematic for viruses for a number of reasons. So one, viruses were so small we couldn't see them, right, with light microscopes at all. So there was just, they were essentially invisible to us. We just knew they were present based off our ability to filter them out with these very, very fine filters. Um, in terms of being able to isolate and grow in pure culture, that was challenging as well, because viruses are obligate parasites. They have to be inside living tissue, so you have to have those living cells to quote unquote grow the virus, and they'd have to be the right type of susceptible cells. And then again, trying to recover the virus from a newly infected organism was, problematic, because again, we couldn't see them. So really, we couldn't really fulfill Koch's postulates for viruses effectively until the advent of the electron microscope in the 1930s. Um, so we're all familiar with light microscopes that are, shining light through your specimen, and then you're using your glass lenses to magnify and view that specimen. And in electron microscopes, you're passing a beam of electrons through your specimen instead. And you get a much higher resolution, and in some cases, somewhat higher magnification as well. But it wasn't until we had the electron microscope that we could actually see viruses, and it wasn't until we could do that that we could fulfill Koch's postulates and really start to scientifically, definitively show that virus X causes disease Y. And you've probably studied electron microscopes before, maybe compared and contrasted with light microscopes. But of course, the big advantage of electron microscopes is that higher magnification and much higher resolution. Um, The disadvantage is that you're always looking at dead tissue. If you're looking at cells, it's got to be fixed and killed. So you're never quite sure if what you're looking at would be representative of a living cell. Um, Light microscopy is much easier, much less expensive. You don't need as much training to be able to do that. You can look at living cells, etc. But for viruses, just in terms of being able to see virus particles, for sure, electron microscopy is key. Um, in my research on viruses, I've really spent a lot of my time doing light microscopy, where we were trying to look at uh, what's happening inside these live cells as they're being infected with viruses. So both can be very useful, but in terms of identification, electron microscopy is where it's at. All right, so lastly, just to finish this up, we already talked about the fact that viruses are not living organisms, but I did want to mention two other non-living infectious agents, just to throw them out here at the beginning of the course. So the first is what are called viroids. You may not have heard of these. These are small circular pieces of RNA. When I say small, I mean about 200 to 400 bases, nucleotides. They are only found in plants. There's no protein coat like you would find in viruses. We'll talk about the structure of viruses in the next chapter. Um, the RNA that composes viroids actually doesn't code for any proteins, as far as anybody's determined. So it's basically just these short stretches of RNA. They get inside cells and they get copied by enzymes in the cell. So you end up with lots of copies of these viroid that can then move from one cell to the next. And this is in plants, through plasmodesmata typically. Um, there are some viroids whose RNA does seem to have some enzymatic activity. They would be considered ribozymes, but that seems to be relatively rare. And so, you might be thinking, okay, well, what's the big deal? It's just RNA. Well, they can actually cause pretty significant damage to their plant hosts, because the viroid RNA gets copied at such a high rate that it's basically tying up enzymes and resources that the cells might need to actually copy their own RNA. So that can be an issue. Another thing that can happen is the viroid RNA can get cleaved or cut into even smaller pieces by the host cell, and then that can generate what are called siRNAs, which stands for small interfering RNA. And these are very, very short pieces of RNA that can actually bind to complementary sequences in the plant messenger RNA and cause it to be degraded. So these siRNAs are basically altering the transcription and translation of host RNAs. And of course, that could be detrimental as well. And the last example of a non-living infectious agent I wanted to mention are prions, which I'm sure you've heard of. And of course, prions are misfolded proteins. So these are sort of like good normal cellular proteins that get folded incorrectly. So in this cartoon, the PRPSC is the bad one. That's these red proteins in the cartoon down here. The PRPC is the good one, the yellow stars. But what will happen is you get that misfolding, and then these misfolded prion proteins can aggregate and accumulate in the nervous system and cause significant neurological damage. So it's pretty scary. And you could ingest this protein in your diet, right? Something like mad cow disease, for example, is a prion disease where you eat some undercooked beef from a cow that contains one of these prions, and then they begin to aggregate and cause neurological damage in the human body. All right, so we're going to end it there, and we will pick this up next time in Chapter 2.